Hi, my name is Lauren Bailey, and I'm going to be presenting my final project for NRC 390E, Evolution and Conservation, on the story of the great American chestnut and its road to revival as a species. So in today's presentation, I'm going to give a brief species background, general history of the American chestnut and the chestnut blight that struck it in the early 20th century. Then I will move on to some evolutionary challenges that are facing the species today, and the current state of conservation efforts as it stands today. And then I will go on to what's next for the species within the next couple of years, and also some long-term goals for various restoration projects. All right, let's get started. So the American chestnut, or Castanea dentata, is a tree species that is native to the eastern United States, as can be seen in this graph I have to the right. And it is a deciduous member of the beech tree family. It's notable for its production of edible chestnuts in very, very large quantities, which can be pictured right above or sandwiched in between those two bullet points. They have very characteristic leaves, and the chestnuts are a vital were a vital food supply for a lot of America. So the American chestnut in America. It once dominated forests in the eastern United States and was utilized by both the colonizers and the native peoples for its valuable natural resources, including timber, which was used because of its strength and rot-resistant qualities, as well as the edible chestnuts that fed millions upon millions, probably billions if we're considering the animal population as well that relied on these really, really keystone species within deciduous forests in the eastern United States. So we don't really hear a lot about American chestnuts anymore because they aren't very present and they basically are non-existent in our forests. And this is because of the chestnut blight brought about by the pathogen C. parasitica, starting in 1904, which was the first pandemic that hit American chestnut species. By late 1950, nearly all adult trees were exterminated. This blight is a canker disease, and it can be seen in this image to the right, and it quickly facilitates the death of infected stems or limbs. But importantly, it does not spread to the tree's root system, so that remains intact. But any stem, any branch that becomes infected eventually dies and thus kills the tree itself. As I stated, though, it doesn't penetrate the root system, which allows for the American chestnuts to propagate from these existing stumps, which was helpful both when these were trees used for timber harvesting and is helpful in proliferating the species to this day following the chestnut blight infestation and infection that is widespread across its entire native range. All right, so let's look at some evolutionary challenges. So in the wild, the breeding population is almost non-existent because they are killed off so young from this chestnut blight. And mature individuals are equally susceptible. They have a maximum life expectancy in the wild of about 20 years, which can seem like a lot, but in the tree's life cycle that is very, very young, they are able to produce some fruit at that stage. They start producing fruit at around 7 to 8 years old, but you don't see a large presence of American chestnuts because they are not allowed really to take hold. And as a result, these surviving populations are incredibly small and fragmented. And this graph I have to the right emphasizes that with something we've talked about in class, the bottleneck effect. So let's dive into some of those challenges that follow bottlenecking. So first and foremost, we have gene flow disruption because you have very isolated populations within the native range. They are still able to proliferate and send up shoots from the root system, but they are rarely able to reach maturity and breed with other American chestnuts. And that leads to a possible accumulation of mutations within the genome. So we could start to see a divergence of these individual populations if they keep proliferating, if they keep sending up shoots, completely genetically isolated from one another. Another effect of this bottleneck effect, aptly named, is the genetic drift and loss of genetic diversity following a loss of a vast, vast majority of the original breeding population. So from the surviving trees, that is already a very small sample size. And then in efforts to generate a resistant plant line, you are taking even smaller sample sizes from the wild. So there is an inevitable loss of genetic diversity in that case. The nature of these experiments is transgenic or hybridization, and then researchers screen seedlings for resistance, and those with resistance are selected, and that maybe one or two seedlings per generation. So from those one or two seedlings, you are generating an entire line of resistant chestnut trees. So from that, you're garnering all of your genetic diversity from these two plants, which leads to inbreeding depression. American chestnuts are diploid, so they have two copies of each gene, which is common. That's what we are. This also means that we can see inbreeding depression pretty quickly because deleterious mutations or deleterious recessive alleles can come to the surface and really start to deteriorate the phenotype of this species over time. And these are really challenging aspects of trying to recover this species following the massive loss that was seen in the 20th century. And I will talk a little bit more about how that can be avoided when I'm speaking about the current state of conservation efforts. So today there are a wide variety of projects currently underway with the goal of rescuing the American chestnut as a species. Methods range from breeding with resistant relatives to generating transgenic lines with introduced resistance loci, or otherwise introducing a gene that allows the American chestnut to resist dying, to withstand infection from the C. parasitica. So one of the first ideas was to hybridize the American chestnut with other chestnut relatives, such as the Chinese chestnut, which evolved alongside the blight and is moderately resistant. So hybrid lines have been generated, and Penn State has been kind of at the forefront in some cases in developing these hybrid lines, and they do show some modulated resistance in comparison to the wild type normal American chestnut. Another way of approaching this is from a genetic engineering perspective. So researchers at the SUNY College of Environmental Science and Forestry have identified a gene in wheat that confers resistance. The OXO gene, which encodes an oxalate oxidase, which allows cells to metabolize and detoxify a primary toxin secreted by the fungus at the periphery of the infected site. So it prevents cell death and it prevents further infection in the tree, and it helps contain it. So it's not preventing infection to begin with, but it's mitigating the side effects. And experiments have shown that this is incredibly effective. So to the right, I have a picture comparing Chinese chestnut, American chestnut, and the Darling 215 and 311 transgenic American chestnuts. And you can see that the transgenic ones, the ones that have this resistance gene, are incredibly resistant to the chestnut blight, and they thrive under these conditions, which is very impressive and very encouraging. There are some remaining challenges, however, and one of the number one challenges is time, as it takes 7 to 8 years for trees to end up producing fruit to get that next generation when you are breeding these to select for resistance. In addition, it is likely that the key to a sustained blight resistance lies in a multitude of different genes and not just one. So further research is needed and is currently underway to identify additional resistance genes, both within the American chestnut genome and without, like we saw in the OXO gene with wheat. All right, and what's next? So blight-resistant lineages will not be available to the public for at least 5 years, but there are still important steps to take at both the individual and policy level. At the policy level, these lineages are transgenic. There's certainly going to be some sort of hybrid, and these genetically modified crops must be allowed to be established within their native range and grown freely. So the recovery of the species really necessitates careful consideration of the USDA, EPA, and FDA to address concerns surrounding these engineered, genetically modified plants, and to really allow the species the opportunity to be reintroduced into their natural habitat and within their native range to fortify this species and to assist in recovery. On the individual level, you can educate and you can plant. Although American chestnuts die within 20 years of planting due to blight, even if you have a 1 mile radius where there's no blight found, it will eventually get to the chestnut plant. It is important to reestablish these small populations because it preserves what little genetic diversity we have left. It increases the sample size that researchers may use to examine the existing American chestnut population. And it also offers a really good opportunity to engage public opinion. And this is essential for reintroduction campaigns, since at a local level, if a government agency is seeking to reintroduce American chestnuts that are genetically modified, it's going to need to be voted on, and it will need the court of public opinion to be in its favor. So the more we educate, the more we engage with the public about American chestnuts and the realities of trying to save the species and the benefits and the massive, massive place that this species held within our culture, the better chance we're going to have to fully recover or moderately recover at all for this species. Thank you so much. Here is when I would pause for questions, comments, or concerns, but I have COVID, so I'm not going to do that. But thank you so much for listening.